Immunoserology and blood banking tests and procedures. Start with the blood extraction. Preparation of washed red blood cells. After centrifugation, transfer the plasma of the patient into another clean test tube. Add as much volume of NSS until the mixture fills 2/3 of the tube. Then, cover it. Centrifuge the samples for 2 minutes at 3,400 RPM. Pipette and discard the supernatant on the waste bottle. Make sure the packed red cells are not disturbed. Then repeat the adding of NSS and centrifugation up to 3 times. Preparation of a 5% red cell suspension. After washing the red cells, transfer 3.8 ml of normal saline solution on 2 tubes. Then add 200 microliters of the washed red cells to tube 1 and tube 2. Then thoroughly mix the red cells using a pipette. Cross-matching using the gel method. Transfer 50 microliters of donor's red cell to a test tube. Then add 1 ml of DG gel salt to prepare 1% red cell suspension. Dispense 10 microliters of donor's red cell suspension to the corresponding well. Then dispense 25 microliters of the patient's plasma into the same well. Incubate for 15 minutes at 37 degrees Celsius. Then centrifuge for 9 minutes. Read and interpret the results. ABO and RH forward typing, slide method. Place 1 drop of anti-A, 1 drop of anti-B, and 1 drop of anti-D on a clean labeled glass slide. Then add 1 drop of 2 to 5% red cell suspension of red blood cells to each of the drop of the reagent. Mix the reagents and red cells thoroughly using a clean applicator stick for each reagent. Spread the mixture over an area. Then gently tilt the slide from side to side continuously for up to 2 minutes. Read, interpret, and record the results of the reactions on each mixture. ABO and RH forward and reverse typing tube method. Place 1 drop of anti-A, anti-B, and anti-D on separate test tubes. Then add 1 drop of 5% red cell suspension on each tube. Then mix. Add 1 drop of A reagent red cells to tube labeled A. Then add 1 drop of B reagent red cells to tube labeled B. Then add 2 drops of plasma to each tube, to tube A and tube B. Gently mix the 2 tubes, then centrifugate for 15 seconds at 3,400 RPM. Then read, interpret, and record test results. Major cross-matching using the manual tube method. Mix 2 drops of patient's plasma with 1 drop of 5% donor's red cell suspension. Then add 2 drops of LISS and mix and incubate at 37 degrees Celsius for 10 to 15 minutes. Wash the red cells 3 times with saline and completely decant the final wash. Then add 2 drops of AHG to the dry red cell bottle. Mix well. Centrifuge the sample for 15 seconds at 3,400 RPM and observe for agglutination. Grade and record the results. Direct antiglobulin test procedure. Dispense 1 drop of red cell suspension into a clean labeled test tube. Then wash the tube with normal saline solution. Immediately add 1 drop of anti-human globulin to the cell suspension. Then mix. Centrifuge for 30 seconds at 3,400 RPM, then examine the cells for agglutination. Grade and record the reaction. Indirect antiglobulin test procedure. Add 2 drops of patient's plasma to a clean label test tube. Then add 1 drop of 5% Group O cells red cell suspension on the same tube. Add 2 drops of LISS. Mix and incubate for 10 to 15 minutes at 37 degrees Celsius. Then centrifuge the tube for 30 seconds at 3,400 RPM. Then resuspend the cell by gently tipping and rolling the tube. Wash the tube 3 times with normal saline solution, then completely decant the final wash. Add 2 drops of AHG to the dry red cell bottle, then mix well. Repeat centrifugation and washing, then examine the cells for agglutination. Grade and record the results. Copper sulfate test procedure. Prick the finger and let the blood flow out. Allow a droplet of blood to fall on the copper sulfate solution. Then observe the movement of the droplet. If the blood floats, then the donor is deferred, but if it's not, then the donor is allowed to donate. That's all. Thank you for watching.